ค Great. Hi, everyone, and welcome to our sixth and final seminar for our Animal Behavior Live seminar series for 2022. So today we have the pleasure of welcoming Dr. Anita Aisenberg. Dr. Aisenberg received her PhD in biological sciences at the Universidad de la República in Uruguay. She's currently the head of the Department of Ecology and Evolutionary Biology at the Clemente Estebal Biological Research Institute, and she's also a researcher for the National Research System in Uruguay. Her National Geographic Award-winning research focuses on studying arachnid behavior from a multidisciplinary perspective, so integrating techniques from ethology, ecology, taxonomy, and systematics. Outside of research, she is also highly involved in outreach and science communication, having participated in science documentaries, a TEDx talk, and has also authored several books geared toward younger audiences. And she's also a member of the Organization for Women in Science for the Developing World, which focuses on supporting and promoting female representation in science and academia. So today she will be giving a talk entitled Where Beauty Hides, Spiders as Models for Elegant Research and Educational Inspiration. So thank you again for joining us, Anita, and you can start with your presentation.
Thanks to you for the nice presentation, and thank you for the invitation to be here. Such an honor for me. And thank all of you for being there. Well, I would like to start by talking about spiders. Why did I decide to focus my research in spiders? And why these animals that are mostly feared can be beautiful and elegant models for research, particularly for those of us who like testing hypotheses related with sexual selection. You know, spiders are fierce, but they also are adored by some of us. As an example, I brought a book by Neil Gaiman. Perhaps you know him because you have seen in Netflix or somewhere else some of the series that are inspired in his graphic models or novels or books. As an example, Neil Gaiman loves spiders too. This book that is called Anansi Boys, there I took a sentence from there, and he says, stories are like spiders with all their long legs, and stories are like spider webs, which we get ourselves all tangled up in, but look so pretty when you see them under a leaf in the morning dew. But there are several particular reasons why so many people decide to work with this animal group. And in particular, they are very special and very excellent models for testing sexual selection hypotheses. And which are some of these reasons? First of all, they are a mega diverse group, and in particular, there are lots of species in the Neotropics, and they have been understudied. So this gives us lots of possibilities for testing and studying new behaviors, describing lots of different things in new species. Also, it's very possible to work both at the field and at the lab. They respond in a very good way to captivity conditions. Also, their developmental periods are in general very brief, so we can follow not only one generation but lots of generations along our lives. They also have very beautiful and different and conspicuous sexual strategies, what makes them also very attractive for this kind of studies. And another special characteristic is that the males of spiders, they transfer the sperm in a capsule. It's immobile. And they have to perform a special behavior that is called a sperm induction, where they deposit the sperm in a very small web, and they transfer the sperm from the web to their palps, that is their sperm transfer organ. In that way, there is a possibility of isolating the sperm and making some nice experimentations along these matters. Also, for those of us like me that have decided to focus on female participation in reproduction and the decisive role that females take in reproductions, female spiders are extremely active, and they are completely decisive when it's time of sperm activation. Yes, they are the ones in charge of the activation of the sperm that is transferred from the male inside their own sperm receptacles. Also, there is a huge variety of mating positions and sexual tactics. We have social species, we have subsocial species, and we have extremely antisocial species, cases with female sexual cannibalism on males, and also cases of male sexual cannibalism on females. So we have a diverse array of possibilities around this very special group. Also, sometimes I think, yes, this is a very good list of reasons, but I would add to that long list that spiders, the thing that they are feared, the thing that they are sometimes understudied, makes us feel like we are rebels when you study them. We are looking at something that only few people can see. And conversely to what you see from the outside, that they are animals, they are hairy animals that inhabit dark places, that we cannot figure out what they are thinking. When you open the door of the knowledge to these animals, you adore them, because they are really like jewels. And it's like Alice in Wonderland. Like you open a door to a fantastic world that is around us, but we cannot see it. So we feel we are rebels, the group of arachnologists studying the spiders. And it has also lots of things to do with our past and with what we can see that is needed in the planet today, to understand that being different can also be a good thing. Well, I will tell you, I will try to summarize in this talk two of my most important research lines that I've been carrying during these decades since I started working with spiders. One of it, it's related with studying cryptic female choice in some orb-web spiders from Costa Rica. When I was studying at the university, most of the books talked, as you will be familiar probably with this, talked about male competition, males that competed for females, and females that were relatively passive in reproduction. And I was not fond of that, and I started reading, and a book that is called Female Control, that was written by William Eberhard in the year 1996. There I found many answers to the things I was looking at. Females are not really passive at reproduction, and there are multiple examples that come from diverse animal groups that shows us that females are decisive when taking mating decisions, not only before accepting mating, but also during and after mating. This is the teamwork in Costa Rica, from left to right. This is the Gilbert Barrantes, me and William Eberhard in the office in the Escuela de Biología at the Universidad of Costa Rica. We have been working for lots of years studying these spiders that I will present to you that are very very abundant in the tropical jungles of Costa Rica, tropical forests of Costa Rica. This is one of the spiders that it's an orb web spider of the family Tetragnathidae. It constructs mostly horizontal webs. The name is Leucauge mariana. Perhaps you have seen it because you can also find it in gardens along Costa Rica and then along many countries in Central and North America also. And this genus, Leucauge, has more than 211 species, but we only know about 2 or 3 species. It has been very scarcely studied. This is another of the species we have worked with that is called Leucauge argyra. In this case, it's very abundant in Costa Rica, as you can see, in the plantations of African palm trees there. And in both cases, males and females clasp their chelicera prior to mating. In both cases, it's the female who needs to embrace male's chelicera before mating occurs. There you can see the female, it's in white, and the male in black points. We found that in both species, both in Leucauge mariana and Leucauge argyra, females presented mating plugs that lower the possibilities of remating. As you can see in the left, this is the external genitalia of the female, and you can see there in white the mating plug. And on the right, in Leucauge argyra, as you can see, sometimes in the left or the right opening of insemination, there can be different mating plugs, and sometimes they have only in one side. You can see there, in this case, they have different mating plugs on one side and on the other one. Well, both species presented these genital plugs covering their external, could present these mating plugs, but in the case of Leucauge mariana, and conversely to what is widespread in other spider species and in other animal species, in this case, the mating plug is formed by both male and female. The male transfers his own substance during mating, but the female needs to add a substance at the end of mating or after mating, and if the female does not add this substance, the mating plug is not effective in avoiding new rematings. And in the case of Leucauge argyra, the mating plugs is formed exclusively by females during or after mating. Not only this, the female starts producing the mating plug during mating in general, and males can get trapped to this sticky substance, as you will see in the video that I will show you in the next slide. So mating plugs in Leucauge argyra can also operate as plugs because the male gets stuck and the female can attack and cannibalize the male after mating. In both species, we found that sexual selection during and after mating occurred. This cryptic female choice. Cryptic, why cryptic? Because for 2 reasons in particular. The processes occur sometimes hidden from our eyes as observers, because something occurs inside the female and it's the female decision to add or do not or don't add the substance, but also because all these processes until the 80s, more or less, they have been hidden by researchers studying behavioral ecology or ethology. Well, in the case of Leucauge mariana, we found that males during mating performed leg pushes. They pressed their own legs, as you can see in the drawing, the male in black and the female in white. They pressed their own legs against the corresponding legs of the female, and the more they pushed and the higher duration of this behavior, the higher probability that the female supplied the substance that is necessary for efficient plug formation. In the other species, in Leucagia argyra, remember the male in black and the female in white, the male performs like if he was playing a song in a guitar. He plays what we call twanging with his third leg. He moves his third leg and presses the silk line that joins him with the female. And the more he made this like twanging behavior and the longer duration he made this behavior, the higher probability that the female supplied the substance and formed the plug. Now we will see a video about what I explained you in Leucagia argyra, where the female, the mating is almost finishing, and the female starts producing this sticky substance. See? This is the female, this is the external genitalia. You can see that like plastic bag that is inflating and deflating, that is hematodoca. And as you can see there, the male gets stuck. I will show it again in some seconds. As you can see, the male is trying to get rid from the sticky substance that appears from the female insemination duct. The male has, on one side he's inseminating, on the other side he has like a special hook that he uses to place himself in the female genitalia. There he goes again. These hematodoca inflations are expected to traduce that the male is inseminating the female. And as you can see, he tries to get himself away from the female, but he gets stuck with the substance that the female produces. And in some of these cases, mating ends with the cannibalization of these males. Well, we have made several research around these topics with lots of students from Costa Rica and from several other places in the world. I chose some examples of this. For example, this work we made in Leucagia mariana, the first species. And as you can see, these species, these are the frontal view of the chelicera. You can see that we can detect several differences between the chelicera of males and females. Male chelicera are much more hairy, and they also present a protuberance near the fang. So we thought that as these chelicera are clasped together prior to mating, we thought that perhaps these hairs and this protuberance were linked or associated with stimulation from the male on the female. So very easily, and you can see with a very simple experiment, we tested this. We decided to create 3 experimental groups. In one of the groups, we shaved the female under the dissecting microscope. Remember that these spiders are like only some millimeters long, but we have to do it very carefully. We prefer not to use anesthesia because this could affect their behavior, so we only place the female and the male and with foam under the dissecting microscope and very delicately perform this shaving. And we had 3 experimental groups. One with shaved females that were exposed to normal males. We shaved all the chelicera of the female. One group where we shaved the male were in the chelicera and they were exposed to normal female, and just as control, a group of normal females and normal males. And we found, as you can see there, that both in the groups of shaved females and males, when we shaved these animals, the frequency of plug formation was much lower compared with the control group. In particular, in the shaved female group. it was like females were detecting that something was not, they were not sensing something, the stimulation from the male received by the chelicera. The males, both males and females, behaved typically in any of the other aspects of courtship or mating, but the mating plague was formed much less frequently compared with the control group. So we expected that there was something that the females are sensing through the chelicera. So cryptic female choice or sexual selection during mating based in cheliceran stimulation could be occurring in this species. Also, we have also sexual dimorphism in some hairs that these spiders have in the sternum. As you can see in the picture, this is a female. So again, we made a similar experiment with Laura and Linda, where we shaved this hair from the females and from the males, and we had a control group and checked what happened when they were very few hair in the sternum when they were removed delicately. And we found that females that presented the shaved sternal seta did not add any substance that is necessary for the formation of the plaque. Just removing those hair was very important for inducing the female to take a decision on plaque formation. Well, now I will tell you about another of my favorite models that it's around South American wolf spiders, where we also found that the sexual patterns were not as they are typically expected in spiders. In general, in the animal kingdom and traditionally, we have all seen that it is expected that in general, due to their investment in gametes, males in general are the sex that goes around, wanders, looks for potential sexual partner, competes for access to females, and females are the selective sex. However, this is the traditional and expected widespread pattern in the animal kingdom. But however, nowadays, we know that there are several exceptions to this great rule. And in some species in which males really made a high investment during courtship, during mate search, during mating or after mating, it can happen that we found new, non-traditional, non-expected patterns for that animal group. And we can find something different. Females that go out, look for males, initiate courtship, and males that are selective when taking mating decisions. This, that it was called sex role reversal, can be total when only males are selective during courtship, mating, or after mating, or partial when both females and males are selective when taking mating decisions. We know probably these exceptions, and I say it like this because we are not sure if they are real exceptions, are probably much more widespread than we have expected. So much more studies are needed around these cases and around new cases and new species. Well, I present you these are the two models we have been working in South America. On the left, you have Alocosa senex, and at the right, Alocosa marindia, it's a little bit smaller, and below you have the typical habitat of each one of them. Alocosa senex is found in open dunes with very scarce vegetation in the coastline, and On the other hand, Alocosa marindia is found also in the coastline of Argentina, Uruguay, and southern Brazil, but in areas with more vegetation, sometimes with exotic vegetation. Both species are nocturnal. Alocosa senex is found in Argentina, Brazil, and Uruguay. Both species are nocturnal, and they construct burrows, holes in the sand, which they cover all with silk to maintain a stable refuge, and they stay there during the day and during the winter, and they become very active during the summer in the southern hemisphere. Well, in these two species, and conversely to what we expected in spiders, we found that in Alocosa senex, the largest species, males are smaller than females. That is very strange for spiders in general, and particular for wolf spiders. Wolf spiders, remember that they don't construct webs to capture their prey. They hunt actively their prey, and in this case they construct burrows in the sand. Well, in Alocosa senex, males are smaller, larger than females. The females are the sex that goes around, wanders around the open dunes, finds a male inside a burrow, and initiates courtship. And both females and males are selective when taking mating decisions. Females prefer those males that present the longest burrows, and males prefer virgin females and those females with a higher body condition. Also, when males they can reject female, and they can be cannibalistic when they reject the females. And they cannibalize more frequently those females that are with smaller and with lower body condition. These patterns are more or less the same in Alocosa marindia, but the difference is that we did not find sexual cannibalism in the smaller species in marindia. These patterns were very new. They were the first two cases of sexual reversal in spiders, and they attracted international media with the cannibalistic males. That was something very new and different, and it was reported not only under laboratory conditions, but also at the field. Now I will show you a video that was recorded by Marcelo Casacuberta, where you will see the courtship and mating in this species, in Alocosa senex in this case. This is the female. Of course, this was recorded under laboratory conditions. The female founds the burrow of the male. That is the male that starts shaking his body. The female waves his front legs facing the male, and she goes inside the burrow. As you can see, the males are much larger than female. The male goes to the bottom of the burrow, and they change positions. The female stays at the bottom of the burrow, the male turns around, and they will perform several mounts and dismounts inside the male burrow. After the mating ends, the male will turn around and he will leave his own burrow to the female. He will go outside, the female will stay inside the burrow, and the male will cover the burrow entrance before going away. Males can be like 30 minutes covering the burrow entrance, and the female is expected to stay there. She will lay the egg sac inside the male's burrow, and she will exit when it's time for spiderling dispersal with the spiderlings on the back, as is typical for this group of spiders. So males will need to construct a new burrow to have the possibility of new matings, and they will have to wait for a new female visit, and the female will stay with the male burrow until she will lay the egg sac there, and she will go out when it's time for spiderling dispersal, and we know that she can remate several times. So around this thing, we started thinking, so why this is occurring with these two species from the coast and it's not more widespread in spiders? One of the hypotheses was related with the habitat. We studied other cases on sexual reversal in other groups, and we found that sometimes these non-traditional behaviors, they occur with species that inhabit harsh, difficult, or extreme, or rigorous environments. And the coastal habitat can be considered one environment of this kind. Why? Because it's subject to very strong winds, salinity, changes in the tide, changes in the temperature between extreme changes in the temperature between the day and the night. So we thought it could be a possibility that this environment was shaping the occurrence of these non-expected sexual behaviors and sexual traits, because we can say it's a reversal in the typical sexual dimorphism pattern in spiders, because in this case males are larger than females. So we decided to study these questions, and our main question was if the rigorous coastal habitat was really shaping the occurrence of these non-traditional sexual traits in the subfamily Alococinae. That is one of the subfamilies of wolf spiders, but unfortunately it had been very scarcely studied. As you can see, Alococinae, we have 132 species that are distributed along the world. And in the particular, in the Neotropics, we have 19 species, of which 17 belong to South America. But unfortunately, we only know just a little bit of the species of this group of species. Also, we had the problem that most of these species have not been described yet, and there's a lot of genital homogeneity, what made the challenge for us and the need to integrate data from molecular data with data from morphology to establish the species boundaries and infer their relationship. So we presented this project to la Agencia Nacional de Investigación e Innovación in Uruguay and to National Geographic, and the project was funded. And this project that is Latin American and Ibero-American project, because we have researchers from many sites in South America and also in Catalonia working with us, from Uruguay, Argentina, Chile, Brazil, from several sites along these countries working with us. And we want to study the origin and evolution of the occurrence of these non-traditional sexual traits in this group of South American wolf spiders, integrating data from behavior, ecology, genetic, and morphology. So for that, we needed to work together with taxonomists, with ecologists, ethologists, of course, working all together to answer this type of questions. Well, this project, it has been like a dream for us, not only because of the interesting questions that new questions that we have found during this process, but also for the beautiful places we have had the possibility to visit. So we choose some sites along South America, and we collected specimens, and we estimated the activity during the night of these new Alococinae species that we didn't know anything about them. We expected to find that if the non-traditional sexual patterns occurred, we would find more active females during the night compared to males. We also made sexual size dimorphism analysis to check in which cases males were larger than females, And for that purpose, we analyzed not only the specimens that were collected during the samples, but also specimens from scientific collections. And we made the limitation of species to infer a phylogeny. All this is necessary for the ancestral reconstruction of sex roles reversal in this subfamily. And we chose, and this is very important, Alococine species that inhabited other landscape, not only coastal habitats. We went to forests, we went to deserts, we went to grasslands to check if the species inhabiting those habitats also had reverse patterns. Here you can see some photographs of the group that went with us to those beautiful places. As you can see, happy explorers around the world. And I will show you some very recent results we have. I don't expect you to check one by one those species, but there you have some of the selected Alococine species from the north and from the south. And you have with different colors there the different habitats that are typical of each of these morphs or species. When we analyzed the data, as you can see with the stars, we found really significant interactions between sex and species regarding carapace width. Carapace width in spiders is a measure that is considered representative of body size in spiders. We really found cases in which there was sexual size dimorphism, but this did not occur with all the coastal species. But what is real is that we found new species from coastal habitats with reversal in sexual size dimorphism. In all the cases that males were larger than females, they occurred in coastal species, but not all the coastal species showed males that were larger than females. On the other hand, we had the possibility of collecting data about some of these species in different localities. For example, Alocosa senex, you can find these species not only in coasts of the ocean, or estuarine coasts, but also in fluvial coasts. And we compared the size of the animals in the different kinds of coasts, and we found that the difference between males were larger than females in both types of habitat, but this difference was striking, was much drastic in fluvial coast compared with the oceanic estuarine coasts. Of course, this project gave us much more questions than answers, as is typical in science, and it's also a challenge to try to find a way to answer these new questions in the close future. Well, this is a study, the master study of one of our students, master students, Veronica, which we mentored with Leticia and Miguel from Uruguay. And she focused on a wolf spider, a very tiny wolf spider, Alococine, that is found in South American grasslands, that is called Paratracocine amica. And through this approach, she found that first of all, she could redescribe the species, and she found that these species from Uruguayan grasslands, she studied the mobility, the nocturnal mobility at the field, and she also studied the sexual size dimorphism and the courtship and mating strategies, and she found that in this case, males and females showed similar body sizes, and males were the courting and mobile sex that initiated courtship. So this spider behaved as it was expected for other wolf spiders, and this agrees with our initial hypothesis that perhaps the coastal habitat has something to do with the occurrence of the non-traditional patterns in this subfamily. We are also studying how the morphodynamics of the beaches can affect characteristics of the populations in these sand-dwelling coastal spiders. We have this is part of the degree stage of Diego, who has been working with Rodrigo and with Omar, and we are comparing the some population traits and body traits between dissipative and reflective beaches in Uruguay. Dissipative beaches are the ones that are preferred for surfers. They have a low slope, they had a broad surf zone, and the sand grain is fine. Conversely, to reflective beaches, we have steeper slopes, they have a narrow surf zone, and sand grains are much coarser. Well, it has been tested in several other invertebrates from the supralittoral in the habitat safety hypothesis that was postulated by De Feo and Gomez, that supralittoral species will have higher abundance, higher development, taxes, and survival and reproductive rates on reflective beaches compared to dissipative beaches, because they are much more protected, among other things, by the steep slope. They are protected from flooding, as an example, and from suffering inundation and many other characteristics that are typical from the dissipative beaches. So we tested this in a spider for the first time, and we found, as you can see in the figures that you have at the right, that all the values were much higher. Carapace width, that is representative again of body size in spider, the weight, and the body condition was much higher in the reflective beach compared to the dissipative beach. But this is the first case that is tested in spiders, and of course we have lots of things to study and to do in the future regarding salinity and regarding several other issues in these and other spiders from the coast. We have lots of plans for the future to study the sexual behavior and sexual cannibalism in other new Alococine species that we found along South America during our field samplings, but we know only just a little bit about the species in other regions of the world and in North America and Central America too. So that is new for spider fans, if you dare to work with these beautiful spiders. And we also are planning to include new locations for the future. We are also would like to study how unfortunately, all our coasts are full of microplastics, as you should know, and we should like to know the effects of these microplastics on these spiders, because they are strictly associated to these areas, and their complete life depends on the sand and on this substrate. So probably these microplastics could be affecting not only their foraging behavior, but also their burrowing behavior, with all the consequence that this could have in the sexual behavior in this species. Also, we are writing a review trying to find more cases of spiders and other arachnids that have these non-traditional patterns that perhaps really they are much more widespread than expected. And we are also trying to understand, and we are mostly sure, that these concepts, this definition of typical sex role, males as competitors and females as selective, have been historically very much associated with our societies. It's stereotypes, and we need to reevaluate and reconsider all these in our animal behavior studies. I would like to end by telling you that we always say that spiders takes us to beautiful places and to meet beautiful people, and it's really like that. That picture is during a field trip in San Martín de los Andes in the south of Argentina. And now we are working together also because outreach and get people and other researchers and politicians, get them to know to what we are doing and why this is important and why biodiversity, particularly in invertebrates, has to be studied because it's mostly unknown. We are working in this project, outreach project, with researchers from all over the world that is called Unexpected Beauty Through the Eyes of Explorers of the Tiny World. I will show you the trailer. There are workshops we are performing, and some of us, nature explorers, visit rural schools in Uruguay, and some of the researchers from abroad, they connect, as we are doing it now, and talk with the girls and boys, and the idea is to strengthen their vocation and to get them to know these beautiful and inspiring animals.
Este proyecto surge como una invitación a salir a explorar la naturaleza y a salir a buscar nuestras vocaciones. Está especialmente dirigido a niñas y niños de escuelas rurales de Uruguay, de nuestro país. En los talleres, no solamente las niñas y los niños interactuarán con científicas de nuestro país, sino que estaremos en contacto vía virtual con investigadoras e investigadores de diversas partes del planeta. ¡Hola, Laura! ¡Hola a todos! ¿Dónde andas, Laura?
Yo estoy acá en mi oficina, y mi oficina queda en Alemania.
Hola, niños y niñas de Uruguay. ¿Cómo están? Soy Nuria Macías, trabajo en la Universidad de La Laguna, aquí en las Islas Canarias. Y nada, yo trabajo con arañas, vamos al campo, colectamos las especies, vemos qué especies son, trabajamos con temas de biodiversidad, descripción de especies nuevas.
Pienso que la ciencia también es cosa de niñas y que no existe un papel o un rol determinado por el género o por la condición social. Por ejemplo, yo soy la primera mujer de mi familia que hizo carrera de pregrado y de doctorado y que actualmente se encuentra fuera de Colombia ejerciendo y adelantando carrera científica.
El talento que tenemos las mujeres también en esta área ha puesto en evidencia que las mujeres somos muy perseverantes cuando queremos algo, muy dedicadas y muy curiosas, y todas esas características son ideales para realizar investigación.
A lo largo de la historia de la ciencia ha habido mujeres pioneras en sus disciplinas y que han sido muy importantes para el desarrollo de la humanidad y de toda la ciencia.
Poder conocer a más personas de otros lugares, de culturas diferentes a mía, y poder compartir un poco de lo que lo sé y aprender con cosas nuevas con ustedes. Espero nuevas colaboraciones con mucho cariño y feliz de formar parte del proyecto Beleza Inesperada.
En el marco de esto conocí a mucha gente, incluso a estas investigadoras de Uruguay que también comparten este amor por las arañas conmigo. Así que quería contarles eso, o sea que hay muchos científicos y científicas que estudian estos animalitos y les invito a conocerlos un poquito más.
En los talleres aprenderemos acerca de estas misteriosas criaturas y sus ecosistemas mediante el juego, porque la ciencia también puede ser divertida.
sumergido en eso, en la investigación, en explorar, que ellos quieren saber eso. Entonces nosotros salimos al patio, jugamos, encuentran algo, ay maestra, ¿por qué esto? ¿Por qué? Y enseguida, ceibalita, computadora, y bueno, y digo, y ahí investigan, sí, siempre se está investigando, siempre, siempre.
Primero me fijo cómo son más los detalles, me fijo si son parecidos a algunos otros bichitos que yo conozca y ahí busco. Si me parece que son eso, ahí está.
Cuando vemos algún insecto afuera de la escuela, agarramos y lo investigamos. ¿Qué come y cómo se reproduce?
Esos grupos de animalitos más pequeños, que a veces son nocturnos, que habitan en lugares más oscuros, que son más desconocidos, como arañas, escorpiones y otros invertebrados, que nos enseñan, entre otras cosas, a aceptar la diversidad y a reconocer que la belleza está muchas veces en lugares inesperados. Es un proyecto que espero ayude a fortalecer y a despertar vocaciones, sea cuales fueren, en las niñas y niños de nuestro país.
Well, I think this is the real unexpected beauty, and this will turn into a documentary film in which Marcelo Casacuberta and Guillermo Kletzer are working with. I would like to end by thanking all the institutions that have supported me for the research I shared with you. In particular, thank you, the Animal Behavior Lab, for this opportunity, for sharing and talking about my love for spiders and our research we are doing in Latin America. I hope to see more Latin Americans talking about their extremely nice research they are performing in several places and through different approaches and anything. I hope you understand now why I love spiders and why I am sure that they make us think about diversity and about being different can be something nice, and about being more generous and better people and share all that we know with the society. Thank you very much.
Great. Thank you so much, Anita. It was such an interesting talk. It's so funny how I can be so scared of spiders, but so interested in a talk about spiders, because I get sometimes when I see them in person, it's like, oh no, a spider, but it was just so interesting to see the pictures and learn everything about those little spiders and sometimes big spiders.
They are loved, really.
Yeah. So we do have some time for questions. We don't have any questions in the chat yet, but we do have a comment from Letitia saying, great project and video. And I agree, it's always great to see outreach, especially with kids, because they get so excited about science.
And they really are not afraid of these animals.
Mm-hmm.
We have seen that in general, small girls and boys, they love all that is related with bugs. They love them. The thing is, something occurs with us when we grow up. And also as researchers, we think that spiders make us like eternal children to us, to the researchers, because we have the same eye and the same interests mm-hmm. that we had when we were very, very, very small.
Mm-hmm. Great. And we do have one question in the chat from Natasha Rossi, who says, thank you for your great talk, Anita. Do we know if there are other characteristics apart from the silk that males are sewing for females that they use to choose males?
Yes. we know that prior to the mount inside the male burrow, the males touch the female. We are not sure if there is a pheromone involved that the males are able to detect and check if the female has already mated with another male or if she is a virgin female. And we also try to test, because remember that in these spiders, all the small spiders, when they are born, they emerge and they get on the back of the mother, and they hang the small babies from the hairs of the mother. So we don't know, and we expected that perhaps the small babies left silk on the top of the mother, and when the male touches the mother, he can detect if she has already mated and if she has already had spiderlings on the dorsum, and that it could be something chemical, something mechanical, but we don't know the answer to that question yet.
Okay, and that kind of leads to another similar question. So I think Natasha was sort of asking about how they choose males, but one question we did have is, how do the spiders detect who is male and who is female? So not necessarily choosing who to mate with, but even just knowing, like especially the ones that are such a similar size and look similar. Is it pheromone based, like you mentioned, or?
Yes, mostly. You must remember that these spiders, they have very bad eyesight. They can only detect movements, and remember that these spiders are nocturnal, so vision is not very important for them, and for us also, because we have to look them at the night with a lamp. And the thing is, in this case, in both Alocosa, the males emit like a pheromone that is volatile, and the female can detect that pheromone and goes out and detects that the male is inside a burrow in a site. The burrow can be closed, with the entrance closed, but she opens it and starts skirting. So we have made some experiments at the lab, and we found that males of this spider emit like an odor to attract the females to their burrow, and it's mainly chemical. And regarding the other question that somebody made, remember that not only males choose among females, but also females choose among males, and they choose those males that show the longest burrows. How they detect this? Because during courtship, the female, remember in the video, she follows the male until the bottom of the burrow, and probably in this way she can detect the length of the male burrow, and if it's too short, she just turns around and goes away. Probably because burrow length is very important then for the female for raising the progeny, because she will stay there.
And she needs more room, I guess, to raise as many as she
room and temperature and humidity conditions that are very important for the eggs to develop.
Great. Thank you so much. And then we do have just another comment from Vera Basas saying, thank you for the talk. Natasha Rossi also has another question. So again, thank you. Oh, wait, no, I think I was scrolling up. Sorry, no more questions. So if anyone does have any questions, you can comment, because the video will actually be available for viewing after the fact. So if no one else has any questions or comments in the chat, we do want to say thank you again, Anita, for joining us and being part, thank you, of our final seminar of 2022. So it's a great honor to have such a great talk closing out our seminar series. And of course, thank you to all the previous speakers and our viewers and everyone who's joined us over the last few months for all of our seminars for this year. And although we are ending our seminar series for this year, we do still have our Congress coming up in November, which is the third installment of our Animal Behavior Live online annual conference. So this conference is completely virtual as well as completely free to both present and attend. And so if you'd like to present or attend, you can visit our website, animalbehavior.live, for more information on the registration and abstract submission. And the abstract submission is coming up quite soon on September 11th. So we are hoping to see a lot of our seminar viewers at our Congress, and we will also be starting our seminar series in 2023. So,
okay, see you there.
Yes, see everyone there. Thank you again.